Thank you very much for your invitation to speak today here. I'm Mireia Valles-Colomer. I'm a researcher now in the University of Trento in Italy. But part of the research I'm going to present here was also done in Leuven in Belgium. And I'm going to talk today about the human gut microbiota and its neuroactive potential together with its relation with our mental health, in quality of life and in depression. Okay, so the notion that the gut microbiota and the brain interact is rather old. It already appeared in scientific literature in the late 19th century when visceral stirrings were proposed as part of the emotion of fear. Some years after, it was found that human neurotransmitters can affect host-microbe interaction. And what I find very intriguing and what I'm focusing my research on is that microbial synthesis of several human neurotransmitters was also detected. Now, in the last decade, there were huge advances in high-throughput sequencing technologies. And this also helped the topic receive notable and increasing attention. So now we know that there are many diseases that are characterized by an altered microbiota composition, including Alzheimer's, Parkinson's, depression, multiple sclerosis, you name it. And in this context, an altered role for the gut microbiota in central nervous system development and physiology, psychology, and even in behavior have also been proposed. So in this context, the microbiota gut-brain axis was proposed as a bidirectional communication system, integrating neural, endocrine, and immune communication between the gut and the central nervous system. The role of the gut microbiota in this bidirectional communication is not limited to modulation of such host neural, endocrine, and immune responses, but also members of the gut microbiota can directly produce and degrade neuroactive compounds. With either beneficial, neutral, or detrimental effects on the host. So this is the part I'm focusing on. These compounds include certain fatty acids, like for example, butyrate, hormones like quinolinic acid, immune modulators, or many neurotransmitters like serotonin, dopamine, or GABA. In fact, it's estimated that 90% of the serotonin in our bodies is actually produced in the gut. So some of these compounds could exert their effects by crossing both the intestinal epithelium and the blood-brain barrier, but that's not entirely necessary, as they could also affect our mental health by signaling to the enteric nervous system, to the vagus nerve, or by inducing the release of other neurotransmitters from intestinal epithelial cells. Okay. So to study the microbiota gut-brain axis from a microbiota perspective, we mostly use metagenomics, in which we start from a sample that is most usually a fecal sample. Then we perform DNA extraction, and then we have two different technologies. The first one is amplification and sequencing of the ribosomal RNA gene, 16S sequencing, we call it. Which gives us information on the taxonomic composition of the sample. So basically, who's there? And this gives us other insights in terms of the diversity of the microorganisms that we have in the sample, global abundances, and you name it. The second approach that we have is shotgun metagenomics, which consists in randomly sequencing the microbiota gene pool. So this gives us a more complete idea of what's there in the sample. So both in terms of taxonomic composition at a more refined resolution, but also in terms of functional composition. So we don't only know who's there in the sample, but also what can these bacteria do, what functions they can carry. And this in terms of gene content and metabolic traits. However, interpretation of this data is far from straightforward, so dedicated tools are needed. Specifically to interpret shotgun metagenomic data in the microbiota gut-brain axis context. So this is what we started with. So as I was saying, I mean, in the last decade, there have been huge advances in microbiota gut-brain axis research, but still analysis and interpretation of such data is hampered by the lack of specific tools and databases. And most probably because of this, most often only the taxonomic composition of the microbiota is assessed. In addition, there are confounders in such studies that are not always taken into account. For example, medication could also influence microbiota composition. And then we might be looking at just the effect of medication and not the actual disease phenotype that we are interested in. And finally, most of the research is still associative, and there is still comparatively fewer research assessing causality. And the studies that do are most often on animal models, with translation to humans lagging behind. Okay, so we started our study by first developing a microbiota brain axis notation framework that would help translate shotgun metagenomic data into neuroactive potential of the gut microbiota. This framework consists of a set of gut-brain modules. We have 56 in the latest version, each following the synthesis or degradation of a neuroactive compound by members of the gut microbiota. These modules were manually curated based on literature and database review, and they follow the KEGG database syntax. So we have, for example, here, one example of such module, which would be the one for synthesis of DOPAC, which is a metabolite that has altered levels in Parkinson's disease from its precursor dopamine. So this pathway consists of 2 steps here, which where we translate EC numbers to something like this. So this is how a module looks, and we have in there the input and the output compound, and then each line corresponds to the orthologous groups to perform one of these functions. So for example, this first line is the one for conversion from between dopamine and dopal, while the second one comes from dopal to dopac. A type separation here corresponds to an OR. So these are different orthologous groups that can perform a function in different bacteria, while a comma is an AND. So these are 2 subunits of an enzymatic complex, and the 2 of them would be needed for bacterium to be able to convert dopamine into dopal. So once these 56 modules were assembled, then we annotated them based on their metabolic pathway, their involvement in different diseases, well, if it was known, and also their potential of both crossing the intestinal epithelium and the blood-brain barrier. This was based on structural predictions or on database data. And finally, we validated them by comparing their distribution in reference genomes. So these are fully sequenced genomes of microorganisms that have been isolated from the human gut, compared to what has been found in experimental data. So if we know that a microorganism produces the neurotransmitter GABA, for example, in vivo, then we want to see a positive prediction in its genome. So if you are interested, the set of modules is freely available in this link here. And for us, what we do was then assessing the distribution of such gut-brain modules in as many as possible gut microbial genomes that we found, to have a first catalog of the neuroactive potential of the gut microbiota. So we have them here, and we found that while some of these gut-brain modules were essentially ubiquitous, so you can see that they are in most, like almost in every reference genome that we assessed. Others were characterized as rare, so the blue ones here on the top, so could only be synthesized or degraded by a very little amount of bacteria. And interestingly, these rare examples contain, for example, modules for synthesis of the neurotransmitters serotonin or dopamine. And only out of, only 30 out of the more than 500 microbial genomes that we assessed and we tested could actually were carrying more than one of these rare gut-brain modules. So you have them here. And we found it intriguing that this contained many pathogens or opportunistic pathogens such as Pseudomonas aeruginosa or Yersinia enterocolytica, which interestingly had already been reported to respond to gut neurotransmitters. So now we know that besides responding to them, they can actually themselves produce these functions. So next, we compared the distribution of these gut-brain modules in genomes of these microorganisms isolated from the gut to their distribution of genomes of free living microorganisms. These were, for example, isolated from soil or from water environments. And we could identify 13 of them as predominantly gut associated. So these are the ones that are colored here. So these, here we have some hints that these would play a role in interacting with a human host, being enriched on it. And these 13 modules, interestingly, included all 3 known pathways for synthesis of the neurotransmitter GABA that are there in microorganisms. GABA is known actually to be produced in large quantities by gut bacteria, where it plays a role in intercellular pH homeostasis and in energy generation. And next, we found that those 13 gut-associated modules actually had a very wide phylogenetic distribution, but they were also very rarely included in plasmids, which hints towards difficulty of horizontal gene transfer of these complex traits. Next, we applied our framework that was then already validated into metagenomic datasets to study gut microbiota and neuroactive potential variation with life and with depression. To do this, we used the Flemish Gut Flora Project dataset, which has fecal microbiome data and extensive metadata for more than 1,000 individuals in the Flanders area in Belgium. We studied quality of life using the RAND Health-Related Quality of Life Questionnaire, which has 36 items. They can be then summarized into 10 scores, 4 mental ones like social functioning or mental health, 4 physical ones like physical functioning or body pain, and there is also a mental and a physical component summary scores that can be computed. Next, depression was assessed by the participants' general practitioners, and we had 121 individuals with a diagnosis of depression. A bit less than half were medicated, taken with antidepressants. So, first step, we assessed what proportion of inter-individual variation in microbiota composition could be explained by anthropometric and gastrointestinal scores as compared to what we were interested in here, so quality of life and depression. And we found that although the effect size of quality of life and depression were smaller than those of anthropometrics and gastrointestinal covariates, then when building a cumulative model, there were also 3 RAND scores, so 3 quality of life scores, namely body pain, physical functioning, and general health, that also entered the model together with diagnosis of depression, meaning that those did not have a completely redundant effect size and explanatory power. So once we knew that depression and quality of life are actually associated with our microbiota composition, even after carefully taking into account the effects of potential confounders, we went on and looked for specific associations between bacterial general and quality of life and depression scores. Interestingly, we found that Ficobacterium and Coprococcus, which are 2 butyrate-producing bacteria, which is a short-chain fatty acids that has beneficial effects for our health, were positively associated with mental and physical quality of life scores, while flavonifractor, which had in the past been associated with depression, was negatively associated. Here, the bulbs that are ticked, labeled with a tick, were validated in another independent dataset, which was the Lifeline Sleep dataset from the Netherlands, that also has fecal microbiome data and extensive metadata for more than 1,000 individuals, in this case from the Netherlands. Next, for depression, we also found 4 bacteria genera that had reduced abundances in participants with a diagnosis of depression. And the 2 strongest associations, the ones with Coppercoccus and Dialister, could also be here validated in the independent data set. And in fact, also hold on after taking into account the effect of antidepressant, and had already been reported in the literature based on smaller case control studies of depression. So we see those 2 genera as the main targets for follow-up research. Next, we applied an approach to stratify the samples of individuals based on their microbiota composition. So in this principal coordinate analysis plot, each dot represents 1 sample of an individual, and dots would be closer together if individuals would have a more similar microbiota composition. The reason why we applied this approach of so an ordination and performing direct hemothermia mixture models on it was that before there were 4 enterotypes that had been proposed, and one of those, the Bacteroides 2 enterotype, has been associated with disease. So that's why we went on to try to further characterize it. And we found that actually here, in participants that had the Bacteroides 2 enterotype, those here, had significantly lower quality of life scores, both mental and physical ones. And this Bacteroides 2 enterotype, the red one here, was also increased, so was found more prevalent in participants with diagnosis of depression. We found in another study that actually also individuals with primary sclerosing cholangitis, and which is a liver disease that is highly concomitant with inflammatory bowel disease, either in its ulcerative colitis or in its Crohn's disease forms, had very high prevalence of that enterotype. So now we know that individuals with Crohn's disease have an almost 80% prevalence of Bacteroides 2 enterotype, whereas in the healthy population it's less than 20%, and those with in those with depression it was around 25%. So we went on to also experimentally to try to characterize this Bacteroides 2 enterotype a bit better, and we found that it was associated with higher levels of inflammation, both fecal inflammation as assessed by fecal calprotectin, but also systemic one as assessed with CRP in blood. It was also associated with higher moisture content in stools, meaning that participants that had this altered microbiota composition had also more liquid stools. And finally, that it was associated with reduced microbial load. Which now we see that actually an increased microbial load in the gut, so a more dense intestinal microbial ecosystem is actually beneficial for our health. And actually the load that we saw in this Bacteroides 2 enterotype was even lower than what would be predicted or expected based on its moisture content. So those individuals that are classified in the Bacteroides 2 enterotype actually have inflammation, but also just less bacteria in general. Next, we went on and applied our gut-brain module framework to assess associations with quality of life and depression. And we found 4 gut-brain modules to potentially be associated, so because they were covariant with quality of life and depression scores. And of these, I would like to highlight the association with DOPAC synthesis, which is the dopamine metabolite that I mentioned earlier, with mental quality of life. So participants with a better, so higher mental quality of life had more DOPAC synthesis potential in their gut. This is actually in line with the literature, as reduced dopamine levels in cerebrospinal fluid have been proposed as a biomarker for Parkinson's disease. And it's this part of the pathways of dopamine transition to dopamine, as I was saying earlier. If there are any questions, please feel free to interrupt me also during my presentation if something is not clear. Otherwise, also feel free to ask at the very end, whatever works best for you. Okay, I was hearing someone talking, but yeah, then I'm assuming it's just background noise. And yeah, then please feel free to ask afterwards. Okay, that's our first project on the gut-brain interaction. And we think that this perspective that John Crine and Tom Dillon wrote on our paper summarized very well the main conclusion of our article. So they were saying that even if we are in the very early days, we might be at a revolutions or a paradigm shift in how we understand and possibly treat depression through targeting the gut microbiota. So we could complement antidepressant therapy with microbiota alterations, modulations. So as a first step in this direction, what we did was setting up a microbiota modulation strategy. In this case, we used probiotics as a potential approach to restore eubiosis in the gut. Probiotics are live microorganisms that, when administered in adequate amounts, confer health benefits on the host. And then we have psychobiotics, which are the probiotics that confer health benefits in patients suffering from psychiatric illness. Then probiotic research has traditionally evolved around bifidobacteria and lactic bacteria. But lately, based on the results of next generation sequencing studies, more and more bacteria, like more diverse one, are also starting to be studied. Butyrate, which I already mentioned earlier in this presentation, is a short-chain fatty acid that is the major energy source for colonocytes. It also strengthens the gut epithelium barrier, it reduces inflammation, and has reduced, butyrate producers have reduced relative abundances in patients with inflammatory diseases. So we chose these Butyriccoccus crucicorum strains, which produces large amounts of butyrate, and it has already been shown to be safe both in vitro and in animal models. And for this, we performed a phase 1 trial for safety in healthy volunteers. So we had 28 individuals that were completed the study and were being included in the analysis. So this was set up as a randomized, double-blind, placebo-controlled crossover study, which means that we had first running period of a week. We asked participants to provide a fecal sample and a blood sample at the beginning of the study. And then they were instructed to take a daily dose of the probiotic, which had 10 to the 8 CFUs of the strain. Or the other group were taking the placebo, which were the exact same capsules, but filled with maltodextrin in this case. And then after a month of taking these, they had a 3-week washout period in which they would not be taking anything. So that was just for us a strategy to go back to the initial microbiota composition in case that would have been altered. And then the participants would take the other product. So we were just giving capsules again. They were blinded, so they would not know if those were the probiotic or placebo, but then they would just take the other compound. So we found that no differences at all in reported adverse events, blood values, microbiota composition. So as you can see here, all samples are scattered around, nor in microbial metabolites. So yeah, especially for adverse events, you can see that none of the p-values is even close to be significant here. So we concluded that administration of butyric is both safe and well tolerated by the human host, and it also does not disrupt gut microbiota composition in healthy volunteers. So in this context, in an intervention study with a therapeutic dosage of a strain in participants with intestinal inflammation can be envisaged. So overall, now there has been more studies on the gut microbiota in anxiety and depression. And these authors conducted here a systematic review, including our article and others. And it's interesting that actually the finding that it's there every time is that there is a decreased abundance of short-chain fatty acid producers in patients with anxiety or depression, together with an increased levels of tags that are associated with inflammation, so potentially pro-inflammatory bacteria. Of course, we can never stress enough the importance of taking confounders into account, such as diet, medication, psychiatric medication was in this case, but also any other medication that the participants are taking, other clinical certification criteria. We know the criteria to diagnose such disease and disorders are not always very fine cut. And also sex, gender variance, basically through hormones levels. The authors here propose a model in which they propose that in a healthy state, there are high relative abundances of short-chain fatty acid producers, low abundances of inflammatory species, and then there are all these neurotransmitters and other neuroactive compounds produced by gut microorganisms that promote a healthy bidirectional communication between the gut and the brain. But then in the context of an inflammatory state, there is lower relative abundance of short-chain fatty acid producers together with an increase in inflammatory species, which then, because of this inflammation, the tight junctions in the epithelium are often compromised. This causes increased bacterial translocation into the blood and a general increase in inflammation in the whole body. So then this communication is disrupted, and there is also an increase in the levels of stress hormones. Of course, I'm saying all the time that this communication is bidirectional. So we think it can go both ways. So either because of an inflammation or unhealthy diet, whatever, in the gut, then at the end, then that triggers effects on mental health, or the opposite. Because of stress, we get systemic inflammation in the body, and this also impacts our gut microbiota. To conclude, in our study, we showed for the first time population-level evidence of microbiome links to mental health. I mean, up to that point, it was more with smaller case control studies. So now we know that, yeah, actually the microbiome is associated with our mental health. Of course, that was only done in 2 populations from the north of Europe. So we are also striving for better representation and to be able to show these in other ethnicities, individuals taking different diets and so on. Next point is that the main microbiota signature in depression and reduced quality of life is characterized by a reduction in the levels of butyrate-producing bacteria. And this is why we then set up this intervention in healthy volunteers to try to increase in the future, when that's ready, in participants with inflammation, the levels of butyrate-producing bacteria in order to counteract this inflammation and return to a more healthy state. And then we propose as a possible treatment strategy this combination of conventional antidepressant medication with microbiota modulations that might help also there. So to end, I would like to thank you all for your attention. I'm also thankful to my former colleagues and all the collaborators that really helped in our studies, together with my current colleagues in Trento. So thank you very much. And please feel free to ask any question. So now, and if something is not clear, also afterwards, feel free to email me. Thank you.
Thank you, Mireia, for this great talk and for accepting our invitation. It was great. So we can start by the questions. I think I will send 2 questions in the chat box.
Sure. Okay, see now. Okay, there is first a question from Leighton. Sorry if I didn't pronounce it properly. Okay, saying that from analyzing the shift imaginary data sets, were you correlating the microbial abundance with quality of life and depression, or were correlating to dopamine, acetylcholine, organism samples? No, it's the first case. So actually, we're correlating for the first part on taxonomic associations, just the relative abundances of these bacteria. And then for the second one, when I'm talking about the brain modules, it was just the pathways. So in sequencing data, we have the pathways that are corresponding to these functions. We would also, it would be very interesting to be able to correlate that to actual levels of dopamine, acetylcholine, etc. But we did not have the data available. And it's actually something very interesting to look at with metabolomics methods. To see whether this is actually correlating with a real production. In bacteria, normally, if they harbor the genes to perform a function, they will be producing it. But it's always a good check that we did in the validation step of our method. But it's of course interesting to see it. What's also it's also cool to see the link that there is in the body between, for example, serotonin synthesis by microorganisms and the one produced by the host. And this would actually help understand better this bidirectional interaction. I hope this is clear. If not, please continue asking in the chat or live, whatever you prefer. Yes, sure. Okay. So next, I'll go to the next question. Saying the data that you collect in the survey must have been very good. It's impressive for participants willing to share all of this information in their use for research. How did we deal with any issues like consent? So what were we doing there? So it's just the head of our lab went to public TV, just saying that we wanted to understand how this interaction works. If participants would be interested in participating, then we would just send collection kits, so fecal sample collection kits, to their homes, and they would just have to fill them, fill also all these questionnaires online. If participants had issues with using online technologies, then we would also help them. And then they would have to bring back the samples to their pharmacies. Of course, they did consent to that. Of course, knowing that all this data is anonymized. So in exchange, they get information on the general picture, but we don't give detailed information per individual because of this privacy. In general, in microbiome studies, as it's not considered as something very invasive, people, and also people are, quite interested in understanding this. We normally have quite good engagement and participation rates. Actually, we are now recalling some of these individuals for a follow-up after 4 years to be able to assess for us the changes in the microbiota in these timeframes. And we are, again, very thankful to our volunteers that are, again, agreeing to participate. What's tricky there in some cases is that, right now, it's actually nice that there are resources to do a lot of analysis on microbiome data with freely available data. Of course, you don't have their private, so identifiable data, but there are resources from the European agency or the US one, or then there is also an R package called Creative Metagenomic Data, whether you have all the information that was consented to share. And this helps us a lot to advance in the field, because then we can, whenever we find something, we can try to whether this is reproducible in other data sets. Okay, so yeah, that's a way. So luckily, our participants are very engaged, and they just they participate. If you have other questions, please feel free to ask. Otherwise, I'll go.
Thank you very much. I think Sophia just put out a question in the chat.
Okay, so I'm going to read her question. So how do you deal with diet and medication variation through metagenomic analysis? Actually, it's a very good point. I mean, for diet, we have, so there are two different ways. One is with food frequency questionnaires. The other is with health diets. So with food frequency questionnaires, we know which is the average diet of the person. So we ask, for example, how often do you eat carrots, potatoes in your diet? What's the amount of food you eat, and so on. But it is still a very tricky to assess diet data in general. So there are now some apps that by saying what you ate, they calculate the nutritional profiles, but it's still tricky. So for us in our large population study, we could not take that into account. So actually, we think that the reason, to go back to the slides, here, that the maximum that we get to explain, so the maximum proportion of individual variation in microbiota composition we reach is still below 3%, is because diet will be actually playing a huge role. For medication, then, but for, so far, the most accurate way to deal with diet is to standardize it. And that's what we did in our intervention study. That was only with 28 individuals. Those were highly engaged individuals. And then we were just telling them to eat specific foods. So these were people working in a hospital, and they were having each of their meals in the hospital canteen. So that was standardized. And that's just the most accurate way that's out there. But of course, there is a trade-off between the size of the study and the accuracy that you can get. And as I was saying, for medication, that's a bit easier. So normally, I mean, either people who took antibiotics in the last 3 to 6 months, most often we just exclude them because these just have like a huge influence. For the rest, what we did here, oops, sorry, forward instead of backward. Here for depression. So what we did was try to take this into account statistically. So we applied generalized linear models, and then we partialed out from there the effects of age, gender, body mass index, Bristol Stool score as a proxy for gastrointestinal transit time, and gastrointestinal disease effects. So that was, for our large population, the best way we had to also then include medication here and see if our analysis could still hold after taking this into account. Okay. I see there are some other questions.
Thank you for your answer, Mireia.
Thank you. Yeah, there is one from Razana. So do you think this new perspective, the relationship between microbiota nervous system diseases, might open a new road to understanding neurodegenerative diseases, since it has a lot to do with cellular stress? Yeah, certainly. I do think so. And actually, especially for Alzheimer's, there is a lot of research going on. And they are also starting to understand the mechanisms that are behind these associations. So yes, we do think and hope really that this can help, because I mean it seems that a link between inflammation and stress and this bidirectional loop that is being caused there has a lot of harmful effects. Okay, so there is another question from Adela. So he's working on something related to mental disorders like schizophrenia or autism. Is there a good protocol to suggest in order to have the good number of samples and collecting metadata? So I would say, okay, these are quite two different cases. For autism, there is quite a lot of research out there. And in that case, what is also tricky for us is to disentangle the effects of genetics, so human genetics and diet. So on that case, it's important to have designs in which, if possible, include autism problems, but also neurotypical children, also their siblings. On schizophrenia, there is also a genetic component, but that's not, there is not that much research in the context of microbiota, so that's not as important for now. But on the number, then on the number of samples, I think the best is to do power size calculation based on the effect size that was found in other studies that have been published and the number of samples that they had. So for example, if there is a 50 case, 50 control study, and then they found something significant with an effect size, but you want to have some like a stronger effect size, then you need to make a calculation to increase the size. There is also a bit of a difference on whether you use 16S sequencing and shotgun sequencing. Until recently, 16S was a lot cheaper, up to 10 times. But actually, recently, prices are really close together. So for now, now the difficulty is more in handling the data. Because with 16S, it's in gigas, whereas with a shotgun, you are in terms of data, so you need more of a computational infrastructure. But well, I know the audience here is computational, so that should not be so much of an issue. But when we have collaborations, mostly with clinicians, then it's something important to take into account, to have a server and, if possible, a high performance computing infrastructure. And then the metadata, the most important variables are gender, age, body mass index, as obesity has also an important effect. Diet, as I was saying, if possible, standardizing, otherwise with apps or food frequency questionnaires. Also knowing intake of medication. So there are some, actually in the website of the Flemish Good Project, there are some links there. And if you need more specific details for a specific study design, please feel free to email me, and then we can discuss this in more detail. Okay. I see another question from Yusuf saying that whether there are any studies on the association between the microbiome and Alzheimer's. Yes, there are some in humans, and then there are also some studies in mice models in which they try to elucidate more the mechanism of interaction. So yes, also in that field there is a lot of research going on, and the field is advancing. Okay, see more of a comment from Nick, which is also nice to see it. So yeah, I think it's very difficult to control data medication, and when asking individuals to supply data, this is very subjective. Yeah, it's completely right. However, one should just be aware of this limitation. It would be interesting to compare control sets with patient reporting. Yeah, the reliance of reporting, that's for sure a limitation. That's why for us, when possible, we also talk with the participants, general practitioners, or with doctors that might have access to their medical history. But of course, that's not always available, and participants might not want to consent with that, which is fair enough. So yes, that's a limitation. I mean, if possible, I mean, we should try to get the more objective and accurate data. But yeah, so for example, here what they were, we also got the remark from some psychiatrists saying that our diagnosis of depression was made by general doctors, not specialists on depression. So even in that is also a limitation that we acknowledge. So yeah, sure. Yeah, okay. I said you agree on diet and medication, so yeah, greatly. Okay, so there is still another question. I'm happy to get as much feedback. Also, yeah, and I'm also happy of to have these questions and all this feedback. So yeah, for the catalog that we've built in this research, this is another question from Olaitan. Is this available to the public? Yes, certainly. And are you making ongoing efforts to update and revise the catalog as you make other discoveries? So this is, yes, available to the public. That's something important for us. So that once we developed this resource, to make it easy for other researchers to also be able to use this. So yes, this public. On the second part of the question, on the ongoing efforts to update and revise the catalog, we certainly want to do this, and we have, and that's about also in GitHub. So I would say that yes, we try to keep updated and to make always updates. But if you at any point read a paper on bacteria are now can also produce such a neurotransmitter using a novel metabolic pathway. So that would also be very important for us. So we make it public, but also are very happy to have any feedback, any ideas on how to improve it. Okay, I see it. There are no. Yeah. So yes. Another question. Yeah, Magdala. So, have you ever thought of creating a database of microbiome related to mental health, something like RDB or Silva, for example, but more specific to mental health? I mean, that would be interesting. We haven't thought about it so far. What I'm going to write down in the chat is this curated metagenomic data resource, which this has metagenomic data sets for also from healthy individuals, but for a number of diseases. And it's also freely available as an R package. And anytime there is a study published, there is a team that, if the data is released as publicly available, tries to incorporate it in this resource. So I would say that, feel free to also access that and so on. And I would say that to start with would also be nice to keep strong focus here in protein measurement data on mental health related diseases. Okay, I see that there are no more questions now. So I would conclude here the talk, and yeah. And if there is anything else, please feel free to email me. Thank you very much.
Thank you again, Mireia. For participants, you can you can subscribe to our channel. I think I already shared the link in the chat box. There you can find a record of this talk. And that's it. Thank you very much, everyone, and especially to you, Mireia, for the great talk.
Thank you.
Thank you very much.
Okay.